will be at the Institute for Advanced Brain Studies at Moscow State University. It is a small institute. It was founded initially as a center for neuro and cognitive sciences in 2016, and then transformed to the Institute of Advanced Brain Studies in 2019. But our group here, a small group, has a large tradition of Russian physiological tools, which I will try to explain in my talk. And our main idea at this session is to show how the fundamental brain sciences and animal models can contribute to understanding of the brain during development and learning. So we'll have talks separated by a lunch, and at the second part of the session, we will have also a general discussion for both days. How can we proceed with the ideas of probable cooperation, both between clinical studies and experimental studies, and also between different international groups. So that's the plan for today. Our first talk will be from Rishi Verma from the Indian Institute of Technology, Madras, on multimodal approach to studying the development of human brain. I think it is an excellent link from yesterday's session.
Thank you. Do I have a mic, or I think I should be on the yeah.
Mic here, and just check.
And I forgot to mention while Ricky started that we have plenty of time for today's talk, deliberately to have more questions and discussions. So welcome, please.
Good morning, and thank you everyone for inviting me, specifically Ludmila, Andre, and Professor Konstantin. The work which I'm going to talk today, I have to say it's not just me. There are a lot of people involved in doing this work. The Brain Center was inaugurated 2 years ago, and the work which we are presenting is, the larger picture of the center is to map the whole human brain. We are working on adult brain as well as the developing fetal brain, and of course these are postmortem specimens. And what today I'll be showing is the process of how we process these brain, all the way from, we have the prenatal ultrasound, then we do the postmortem MRI, and what we do with histology, and some data which we have gathered in the last few years. What you see here is, and I would like to give a little background. To process post-mortem whole human brain, there are different challenges. And one of the primary reasons why this has not been achieved at this scale before this is because there's a lot of engineering and tools which have to be developed to process the whole biological tissue as one particular, as a single block, which I will show you what we do. We start this process. So in today's talk, I will talk about what are the challenges in studying the organization of the human brain, and specific to the fetal brain, what has been our approach, and the current data set. What I'm going to show you is from the 14 to 27 gestational weeks. Soon after, we get the consent, within 6 to 15 hours, we obtain a postmortem MRI. These MRIs are done in the clinical setup, and I will show you, yes, it's great, we are able to get these MRI with 3 Tesla in most of the cases, but we do have our own challenges, given that the time for collecting these data is 30 to 40 minutes. Once the MRI is obtained, We leave the specimen in the formalin, the 4% paraformaldehyde, for 2 weeks, and the brain is then extracted. Once the brain is extracted, as you see here, we do a surface scan of the brain, and this might seem like an overkill for a lot of people thinking, why are you doing a surface scan? But what we do here is we generate 3D models of each brain. And when we do that, that allows us to create a cast when we freeze this entire tissue to keep it this in close alignment, the way the brain was sitting in the skull. And we use MRI as well as these surface scans. Then the brain goes through the cryoprotection. I don't have to talk about it, but if anybody wants any detail, please ask me. We put it through graded sucrose from 10 to 30%, and then we freeze the entire brain in isopentane dry ice bath at -80°C. Following that, the brain goes through the cryosectioning, and what you see here is we don't use the traditional brush method to hold the tissue and put these sections are 20 micron thick, and at times we do take 10 micron, but we use what is called a tape transfer technique, where the entire tissue is stuck on the tape, and when to peel the tape, we use a UV curing station, which uncues the tape. The benefit of doing this is we are able to get close to thousands, and in adult brain up to 10,000 of these serial sections with minimal tissue loss. Imagine that doing that with holding a brush and free floating it. It's limited. You can do a few hundred, but not at this scale. Once that is done, the another thing I wanted to highlight here is MRI is a first reference, which allows us to put all of these histology sections back together when we do the 3D reconstruction. But the second thing, what we do here is before we take any of these tissue, we do a block face imaging. This is the UV curing, which allows us to peel the tape. The classic staining which we do is Nissl, HNE, myelin, and a few immunohistochemistry based on the question which we are asking for that particular age group. Then all of these histology sections then get digitized. And then because, as I mentioned earlier, our bigger goal is we want to map each of the brain regions. We then do the atlas annotation. And because now you have the digital data, all of this is done through a custom design tool. And then we put all of for example, the data which I will show you today here from 20 week, will have 2,000 of these histology sections digitized. They are then put back together, which then gives you a 3D reconstructed brain, but more importantly, at the cellular resolution. Now, why are we doing this? So if we look at the developing fetal brain atlases, the two atlases, which is currently the so-called gold standard, one is, of course, the MRI atlas, but the MRI atlas starts from 20 week onwards. And this is from Golipur et al. From the Harvard group, and it is quite detailed atlas, but at the same time, MRI has its own challenges in terms of resolution, and we don't have atlases between 12 weeks, the samples which we have, all the way up to 20 weeks. The other one, which is the histology printed atlas, is from Baer and Altman. It's in 5 volumes. Anyone working in the developing human brain would be aware of this. These are quite detailed one. The only challenge there is each brain specimen would have maximum of 20 sections. So you cannot study the whole brain with just 20 of those sections. Given that MRI lacks resolution, the histology current available atlas do not have, they're covering the entire brain region. That was our approach. How can we do both, where you 3D reconstruct and you get the entire volume? And also the processing which we do allows, or rather prevents, tissue shrinkage to a larger extent. Hence the 3D reconstruction is much more feasible in this particular case. The other one, which has come in the last 3-4 years, is the Digital Atlas by Allen Brain Institute. And the developing brain, they have put 3 brains, 1 hemisphere each. You can navigate through these, atlases. Each of it has, I think, 80 or 100 sections, and that can tell you which region you're looking at it. You can even zoom in and look at the cellular level details. What we have done in the last 2 years, we have processed brains from 12 weeks onward all the way up to neonates. And of course, as we go anywhere from 27 gestational weeks to later stages, the number of samples are less. But in the earlier stages, we definitely have 3 to 5 specimens in most of these age groups. And each of these brain, you are looking at anywhere starting from 1,400 to even 6,000 sections at this detail. Currently, we share this data with our collaborators. It's just access through your Gmail account, but we are working on making it as an open access as we move forward, and there's a lot of work involved doing so. So when each of these histology sections are digitized, what we work on in each of the 2D images, we do the atlas annotation, we look for different types of cells, we do different immunohistochemistry, we also try to classify the developmental stage. Connectivity, we do myelin, but at 27, 28 weeks itself is quite, most of it is not myelinated. And then what we are currently working on is doing morphometric. Because we are able to do the 3D reconstruction, we should then be able to measure different regions within the developing brain with histology, MRI, as well as the prenatal ultrasound. And that, I think, is an important part for anyone working in the clinic to look at it if there is normal, abnormal, and I think that's how we define most of these parameters. When it comes to putting all of it in 3D, we do the MRI registration so that the alignment is accurate. We do the block face imaging, 3D reconstruction, and of course, then we look into volume analysis and the morphometric, compare 2D versus the 3D measurements. This is a short video which shows you, okay, how do I go to the, okay, now I started. This, what you're seeing here is an MRI taken on 3 Tesla for 14 gestational week brain. As you could see in the middle panel, you can access the data in all 3 planes. This is the same brain which we did a histology, reconstructed. We sectioned in sagittal plane, but because we were able to do the 3D reconstruction, now you can see in coronal as well as the transverse plane entire brain, and then you can click on the region of interest, and that will take you to the detailed higher resolution. What you see here is our user interface, where if you go to any particular brain, you will look at all the modalities in block face, BFI, as well as this. And when you click on in the image, whichever you are interested in, this video will show you. And as you see, it's a whole brain. In the sagittal plane, you can see the cerebellum, the brainstem, even a bit of spinal cord. And once you digitize this data, this is the resolution at which you are investigating all of these samples.
What is the resolution, actual resolution, 20x, 40x?
No, it is 20x, but in terms of pixel, it comes down to 0.5 micron per pixel in plane resolution. And many times when we have to share the data, especially if there's a large section, we do have to downsample it. But based on the requirement, you can change the resolution of these. And this is the same brain, just to highlight. The bottom panel is the MRI of the brain done. And as for anyone, even if you're not in the field, you can appreciate the contrast and the resolution of the same brain and the details which you could get with histology that won't be possible with the MRI. This is another example to show how we go about doing the atlas annotation. What you see here is a 22-week specimen, and the different colors here represent the different regions. We are working with few collaborators to develop the ontology and work towards it. One of the reasons doing this in developing brain is a lot of time the nomenclatures are used comes from the adult brain, and at early stages most of those regions are not even formed. So this is quite a detailed work, and I thank collaborator Mihal, who is working with us on this. Once we do all of these region marking, we then, this is again a video to show the stack alignment of the same 22 weeks. And the reason I chose, this is a brain which we processed in the coronal, and I'm guessing most of you are used to seeing coronal section. This is, you're looking at the histology, the images from the front of the brain to the back of the brain. And I hope you can appreciate the details and the loss of tissue, which doesn't happen in our pipeline. And you can literally look at cellular level the way you would see into MRI, and rather with a much higher resolution. Since yesterday we had this conversation of 21, 22 gestational week, I wanted to highlight we have done like 6 to 8 brains in this age group, and as you would see, the brain is so underdeveloped at many levels. By 27 weeks, we do see quite a developed brain, which looks closer to what you would see with adult at least. But I was very impressed and surprised that when you said even the major sulcus are not visible at this point. So I'll just move forward. This is the same brain with different modalities, where you see the postmortem in skull MRI, the block trace image, and the serial sections with the nissle. Then there is a whole big computational team which works on putting all of this data together to do the 3D reconstruction, but it's a lot of work involved in making sure that you can look at the cellular resolution, and those are done by using all the annotated sections where we mark region, they are all put back together. And this is something what we want to release it as an open access data. Talking a little bit about MRI, given a lot of people here do work on it, we do take postmortem MRI. What you see here is the number of specimens which we have done in the last couple of years in different gestational age. The one highlighted in the rectangular box are the ones which were taken with 1.5 Tesla, and hence the resolution is not that good. What we are finding, and this is something I would like to discuss with the group, is we are able to get MRI atlases postmortem. It's pretty good, but when it comes to doing atlas segmentation, region segmentation as detailed to what one would compare with histology, we do have few challenges. In 2D, we are able to mark the regions quite well, but not in when we put the entire volume. And also given the fact that for early gestational ages, especially anywhere from 14 up to 22 weeks, no atlases exist at this point. So we are trying to do this with our histology, and given the size of the brain, the clinical setup where you cannot record MRI for 4 to 6 hours, we are limited currently with 40 minutes to an hour. This is the signal to noise ratio which we get, but we are definitely keen to expand this further and try to get a higher resolution. This is just to highlight what we have done so far. We have done most of the brains in this age group. We definitely have few neonates as well as 27, 28 weeks, but the number of samples are less because, as you know, with the good perinatal care, now we don't get those many samples, which is actually a good news in a different way. I just want to share what we have been looking in these data. And one of the things which we are interested in looking at, what are the different anatomical histology data in this human specimens. To start with, again, I don't have to say much of details. We are looking at neurogenesis, the migration, the differentiation of these neurons, of course, the laminar organization, and at early stages, as you would know, the 6 layers cortex is not formed. We label it starting from ventricular zone all the way up to marginal zone. We are also looking at, and this is something at early stage, to look at the thalamocortical connections, corticocortical connection, and then see how the pruning happens at later stages by trying to do automated cell counts as well. This is just an example to see my background being in the vision science. I'm interested in looking how the visual area, especially the primary visual cortex and the LGN develops from 14 to 24 gestational weeks. As you could appreciate, these are all on the same scale. At 14 weeks, the cortex has these layers starting from ventricular zone to marginal zone. As you move further up, you do see the changes in the intermediate zone, but by 24 weeks, even in this cortical plate, the 6-layer cortex is not very visible, and I will talk a little bit more about it in a minute. What I've shown here in this panel is we do different immunohistochemistry. Again, these are based on the region, what we are investigating, and also the type of questions which you want to look at. But the benefit here is having the whole section, we can do comparative analysis. Now, anyone working in the visual development of vision in humans or animal would know this. In humans, the LGN has 6 layers, and you get these projections into the layer 4C of the primary visual cortex. And to look at this, I started with the MRI in the 14th gestational week. What you see on the top panel are the 2 arrows, one pointing to the early stage developing LGN, and of course, this would be your occipital cortex. And we do this, sorry, I'll just go back a second. We do the annotations very carefully to mark these regions, and especially in the subcortical region, this requires really looking carefully of because these regions are very in early stages, and I have to say the ones who do it, it's a lot of work marking each of these regions very in detail. And if I take a section from the occipital cortex at 14 gestational week, as I showed you earlier, these are the layers. The subplate and cortical plate boundary is over here. And we do GFAP staining in most of our developing brain. And as you would see from the ventricular zone, you see these radial glial fibers running all the way up to marginal zone. We also do Neuen staining, which stains for the post-mitotic cells. And as you see at this gestational age, there are which are presented like a little black dots. There are very few in the lower cortical plate. As we move forward, the LGN in this particular gestational stage does not have any laminar organization. More importantly, the cells are all small, round in shape, and the processes which we could see through only with HNE, you start seeing these processes coming out of the lateral geniculate nucleus. But as we move to the 21 gestational week, we start seeing the development of calcarine cell a bit more clearer by this stage. The cortex is, of course, developing cortex is much bigger by this stage. You see the ventricular zone, same cortical plate marginal zone. When we do GFAP at this stage, we start seeing maturing astrocytes, which are present not just in the subcortical region, but also in the developing cortex. When we did Neuen with this, again, you could appreciate the difference compared to the previous one. This is a much larger band of post-mitotic cells, which we see at this stage. When we look at the LGN, once again, the cells are a little bigger, but we only see little early stage of magnolayer, which is visible, and the processes coming out of LGN are a bit more dense. We then decided to do specific stain for looking at these optic radiation projecting to the primary visual cortex using parvalbumin. This has been done in the adult marmoset brain quite detailed manner. And once again, we were able to show this, what you see in the bottom right panel, that you start seeing these well-labeled fiber tracks coming out of the LGN. We want to compare, for this particular brain, we did have 7 Tesla DTI as well. Currently, this analysis is in progress, so I will not speak much about it, but I want to do this at different stages in the developing brain, all the way up to the neonate stage, and see how it matures and how these connections are formed. If I look at the 27 gestational week, few things, the calcarine sulcus is much more mature. And if I look at the cortex here, we can really see the 6 layers being visible here. The LGN, even though it's in the sagittal plane, which is difficult to appreciate to what normally you are used to it, but you can start seeing the laminar organization and the layer which starts forming at this particular stage. Also, if I zoom in, we start seeing the V1, V2 boundary also at 27 week. And hence I said, by the time we are seeing 26, 27 gestational week, most of the brain looks much closer to what you would appreciate in the adult brain. So this is where we are. We are really looking to map these neurotypical brains from 12 week onwards. That's the youngest samples we have at this stage to the adult. Having said that, we also have worked and processed a lot of acquired, developmental disorders, including the hydrocephalus. Yesterday, we had a talk, holoprosencephaly, agenesis of corpus callosum, etc. at the same time, and we are comparing those with the neurotypical brains in this age group. To summarize, we have this platform where we can do histology as well as put it back just like MRI in full volume. And that's the gap, if I have to say in one sentence, which we are trying to close, is getting this cellular level details of the brain and putting it back for the entire volume of the brain. And this platform allows us to study using different modalities. And we really think MRI is great to getting the large picture. But if you want to look at the pathophysiology of any of these conditions, etc., we do have to go at cellular level. One thing which we would like to do, and we are working towards, is doing the molecular work in these samples, which is quite challenging given that these are postmortem samples. With this, I would like to thank our funding bodies, the director of the Brain Center, Mohan, and our collaborators. And few of our collaborators are sitting here, Paul and Adil. Paul has been working with us for 2 years. Given his detailed neuroanatomy background, it's been very helpful for us. And as I said, the work which I've presented, currently the center has close to 100, 120 people. So it's not just me, there are a lot of people working on it on different aspects. Thank you. And in the last 2 years, we've done a few papers, all our methods paper are out. We are hoping to get the few of the histology neuroanatomy papers in the coming years. Thank you.
Questions, please. Let me start. We have plenty of time for questions.
Okay.
What about immunohistochemistry? You mentioned that you do it along with the histological staining.
Yes.
How do you perform it? Do you have neighboring sections that they can align it?
Yes. So when we section, we usually do 3 series. So one NISL, one for IHC. We don't decide which IHC in that series. Third one is HNE. Now, myelin is no point at early stage. So these 3 things. When we start with IHC, we have, classically we do NUN, GFAP, and few times now synaptophysin also in the developing brain, paravalbumin, calbendin, calbendin. But once the brain is processed, then we decide what all do we want to do in this particular brain, and we do the neighboring section.
Do you stain them on tape also?
Like these are the ones where our immunohistochemistry is not on the free-floating sections. These are 20 micron thick, and these are slide mounted. And hence it took us a long time, and I would refer to that paper, how we have optimized it. So there were 2 challenges. A, these sections are minimum 2 inches by 3 inches, whole brain sections. And when you do IHC on a slide mounted, it's much difficult than the free floating one. Third, these being the fetal brain, very high in water content, the tissue is very fragile. So many times with the immunohistochemistry initially, we were having a lot of tissue damage during antigen retrieval process. But now using boric acid and more alkaline pH, this has really worked.
So can you do the slide mounting and staining on slides up to what age? How large?
We are even done on adults, which is 6 inches by 8 inches.
How do you cover them with antibody solutions or wash?
No, so antibody being super expensive, we do pipette it, but then we use the paraffin, cover it to prevent, and of course the pap pen, just to make sure it doesn't dry out. That actually we have optimized, but I have to say it's a bit of a slow process when it comes to that section. But yes, and we have been doing this for 1 year with the adult. Adult brains are a bit more robust with doing immunohistochemistry. Even without antigen retrieval, it's much better. But developing brain A, which molecules are present, protein molecules, it's also not well mapped. And here antigen retrieval plays an important role, given that these were in the fixated.
Sure, yeah. It's easily used in the embryonic brain. More questions, please. From our group, which uses very similar techniques. Any questions?
And I wonder if it is possible to highlight special cellular events associated, for example, with the brain expression or some structure formation. I mean, just to improve early diagnostics of different developmental disorders.
With the yes. So we are doing. So I have not presented it here. We have processed in a similar manner holoprosencephaly brain. Now, there are a lot of things already known about it, but it's never been seen in the entire volume. And that's a condition which goes both anterior, posterior, dorsoventral, lateral. And we are able to show at cell level where the things looks like it's fused, but at cellular level cells are looking much more normal. We have done few of the immunohistochemistry and trying to assess. It was an A lobar. Yeah.
More questions? Please.
So how do you think, is it possible to make fluorescence?
We are doing. The challenge was antigen retrieval. The same thing was not working that good with fluorescent, but we have done, there are 2 or 3 markers like calretinin and even Hopex. Those have worked well, but there are a few we are still optimizing. The only thing is the cost is much higher with the fluoro one, but we are working towards it. There is another limitation. To digitize this data, the scanners which we use, they do not have the fluoro options. And they can do it, it's just something on cost we are working on. So I know a lot of people have been asking us about fluoro, especially we would like to do 2 or 3 markers at the same time. We have got successful with couple, but the rest we are optimizing. Yeah.
What scanners do you use?
These are from Huron, only one company in the Canada.
Very good one.
Yes.
But they do also fluorescence.
Yeah, they're just charging us a lot. But that is something we will be doing. At the moment, we just have the new fluoro microscope and we take small regions with that. Yeah.
What about analysis? There is so much data, and I'm particularly interested, you will probably understand from my talk, in the heterochronic maturation of different components in the cortex, subcortical areas, which would be revealed even with the ordinary histological staining, but with NOAN and other markers too, which are extremely important. So we need
at this point
involvement of neuroanatomists.
Yes, and that is something I'm really looking for. We have few people at the center. There's somebody looking at the development of inferior colliculus pretty much to a larger extent from 12 weeks to 27 weeks. Superior colliculus, LGN, I'm looking at. Yesterday I was talking to Adil for hypothalamus. We're really looking for people who are working in different regions. If you're interested to look at it, it's very straightforward. We share the data with you, you look at it, and we go from there. So, you tell me which region you want to look. Because one thing we are quite mindful of, we have generated this data, new knowledge needs to come out of it, including the clinical application. And hence, we are not doing just the neurotypical brains, both the developmental disorders along with it. And so you tell which region and we are okay to absolutely fine, rather than the data sitting with us.
And in terms of initiating the discussion about collaboration, probably the brain samples from preterm born
Yes.
children would substantially enhance
Oh, big
the
time.
collection.
Yep. We have processed, we have 4 or 5 neonate from 28-week onwards. We have processed just one when I was heading out, which is 39-week, had severe asphyxia, and you could see those changes at cellular level there. But again, we would require an expert who would say, look for these markers to really show whether that was an acute one or how it happened, because I think that baby survived only for 4 hours. So those are the things we are really looking into. So this collaboration where, you know, cross-border is the challenge on its own, but we are very open to get the brains and process it because the platform is pretty much set to do any of those work.
I think what they are doing is a fantastic perspective for the future. Any more questions, please? Then thank you for coming.
Thanks.
I will talk in English, but I will have critical slides and some slides in Russian, and in critical points I will switch to Russian for the audience to take it easier, not from the online translation. While the starting, Richard, where is Richard here? I wanted to tell that what you are doing resembles me so much the techniques through which I passed from middle of 80s. I started in 1984 or 85 in order to look for genes which are specifically expressed in the nervous system. I started with the genes which my colleagues asked me to do because I was working in collaboration with the Institute of Molecular Biology, and they were interested in proto-oncogenes. The genes that were just discovered in '84, and it was known that they do something in the organism, though their viral genes are originated from them, and it was not clear what do they do. The hypothesis was that they are expressed during development and do something important in cell proliferation, and when the viruses catch them, they break the regulation and then start to produce pathological proliferation for tumors. And the challenge was to see the expression of these genes. For these genes, there were only DNA probes. There were no antibodies. They were just discovered. So I started doing in situ hybridization on the developing embryos and neonatal animals, rats and mice. And the problem was very similar to yours.
Yeah.
It's a large size. Then it consists of many organs. I was doing cryostat sections of 20 microns. They fell apart.
Yes.
So I started using the tape to cut the block on the tape.
This was in 80s?
85. And the most challenging was, I must tell you, that when you dry them, the tape starts to curl.
Yeah, I think in 20 years they will improve the tape. That's all they're done.
Yeah, I was using the usual tape, by the way.
Okay, yeah, these are comes with a special glue.
And the second challenge was that I was doing not immunocytochemistry, but radioactive in situ hybridization. So you have to apply the radioactive probe and then expose it to the film.
Yep.
When it is dry, it again starts to the film. So there were many challenges of that. Then we switched to, and I was using parafilm as well, of course, for to decrease the amount of probe for that. And we started.
Recording in progress.
Hmm? No, nothing of that.
We are rather thinking of doing ISH. It's just, it's not moving the way we would like it to move as of now. And raising these probes for human specimens is not.
Sorry.
How many times between correction sections and
Okay, so I will say from the time we start cryosection to do the entire staining is for a developing brain, it's like close to, I would say, 4 weeks. And then digitization, you can add another month. But anything like that is like much longer than that. But staining doesn't take long. Immunohistochemistry is a bit more longer, and also we don't want to do bulk, just to given the cost. So yeah, a brain from the time we start and then digitizing developing brain, you're looking at 2 months, which should be okay. Yeah, but larger brains, longer time.
So I tried to put in this presentation something which makes the further step after you in the transition from our first day to our experimental models in the second day. What we are interested in, first 3 slides in the Institute. I told you that the Institute is young, but we have a school, physiological school or neurobiological school in Russia, from which we come. It is nearly 90 years of different generations, and we are interested in functional systems. Just as an introduction, functional systems are distributed elements which have distant anatomical localization, but cooperate towards adaptive results achieved by an organism. So viewed from this point of view, even the most complex phenomena of high nervous activity and adaptive behavior can be studied from this point of view. There are many laboratories in Russia working in this direction. In the West, it is known from similar approaches from the concept of Donald Hebb, neuronal assemblies, distributed systems of Vernon Mountcastle, global mappings, Gerald Edelman, neuronal coalition, etc. And here is the quote of from the work of Wolf Singer and Andreas Engel, describing the very similar approach developed also in the Western neuroscience. Looking for the adaptive behavior and cognition. Another aspect which we do a lot, we work a lot in comparison of different species. We are interested in the issues of evolution of the nervous system, differences in the ecological conditions which produce different sorts of neural organization of behavior. So we switch often during winter period with the techniques of laboratory work, with the optical imaging of neuronal activity, mapping of the brains, while our summer work is also carried out at the biological station of Moscow State University using the different wild species. And there will be a talk by Olga Ivashkina today. She will describe this line of research. Now, what I will focus on is brain before and after birth. And two main messages of my talk will be, there is no sharp borderline between the brain before and after birth. Не существует жесткой границы между мозгом до и после рождения. However, there is a sharp border between development and learning. Однако существует отчетливая граница между развитием и обучением и пластичностью. Каким образом это может быть? How this can be? This is something I will try to describe and explain. And my talk will consist of three portions, three parts. Theory, in which I will describe the theory of functional systems and its continuation into the theory of systemogenesis, development of these functional systems prenatally and postnatally. Methods that we've been developing, and I will tell about immediate early gene imaging, which we developed, and transparent brain. This is especially for Richard. We discussed about optical clearing and using that. And also experimental models in animals. So how can animals help to study the development of functional systems and perinatal learning? That means prenatal learning for some functional systems and early neonatal learning, this transition before and after birth. So first, theory. Functional systems theory was developed by a student of Pavlov, Petr Kuzmich Anokhin, who, after leaving Pavlov lab in 2029, received his department of physiology at Nizhny Novgorod University, and he was dissatisfied with the theory of reflexes, conditioned and unconditioned reflexes of Pavlov. He was very attentive to data of neurobiology that were accumulating at that time, and saying that both from the embryology side, from the development of behavior and studies, neurobiological studies of development of behavior, from the adaptive behavior of animals, and from the processes of compensation of organisms, which involve plasticity, a reflex cannot be an explanatory principle of the operation of the nervous system. So at Nizhny Novgorod, he started 4 lines of research. First, he started comparable to Pavlov laboratory to study the conditioning in adult animals, in dogs, in free behavior. That means in Pavlov lab, dogs were fixed. In this type of dynamical studies, they were active. They were making, for example, behavior choice between left and right, doing operant behavior with all the physiological recordings that were available at that time, of the salivation, of respiration, heart rate, and later on EEG. So that was the dynamical studies of conditioning. Embryo physiology of behavior. the whole range of developmental studies, including the interaction between development and learning. So they were doing the Caesar section and preterm birth for guinea pigs, for example, and looking whether it is possible to make conditioning in pretermly born animals. How early the learning can start, whether it can start before the birth. And many experiments on the compensation, which is issue of plasticity, like using the heterogeneous anastomosis, where what central part of one nerve was crossed to the peripheral part of another nerve, and the studies was how the organism can adapt and compensate to these unusual afferentation or efferent impulses. Or transplantation, for example, of the tenders of the extenders to contractors, so that the animals, instead of, for example, putting the leg during locomotion, was raising it, and to see again what happens with the central integrations to finally receive the adaptation to normal locomotion. And from this set of studies, the concept of functional systems, contrary to the conditioned reflexes or unconditioned reflexes, came from. As it is said here, it was from the very beginning appeared as a self-organizing entity with feedback on the results of the adaptation. The concept of feedback was introduced in 1933, and it was called re-afferentation, reverse afferentation. Later on, by Norbert Wiener, it was called a feedback. An important part of this was to see how such functional systems, which are bringing the adaptation from the cooperation of different parts of the organism, are assembled during individual development. Since the composition of functional systems cannot be defined by any anatomical localization, it is a distributed system. How it can happen that many parts of such system during the individual development came together to be assembled at a particular time point of the development, adapted to the ecology of this particular species. Many of them have to be mature and consolidated just before birth, but other functional systems develop later on ontogenetically to the particular tasks of the developing neonatal or young organism. So there were three questions about this process, which was named systemogenesis. Genesis not of organs, organogenesis, or tissues, tissue genesis, but of distributed adaptive functional systems. How such integrations are formed during evolution, when they are innate. How such species-specific functional systems are assembled during individual development to anticipate the ecological requirements. We can call it the primary systemogenesis. And also how these functional systems are adaptively modified by individual experience, learning, plasticity in the lifespan of the individual organism. So we can split the theory into three parts. And one, which was mostly development, was about primary systemogenesis. There were four principles that were studied experimentally in many species, including human neonatal and preterm born babies. They have studied these in 1950s, for example, in preterm born babies of 500 grams, and followed the development of suckling functional systems for a number of weeks. So that four principles are here. I cannot go into detail right now, but what is important is that besides the primary systemogenesis, which results in the consolidation of functional system, and this is the subject to natural selection. Starting from the moment when this functional system is consolidated, it can perform behavior, its adaptive functions. So if there is any mismatch in the results of this adaptive function, the system enters the stage of plasticity and adaptive modifications, which is learning. So besides primary systemogenesis with the developmental outcome of the adaptive result, there is a stage usually for most of the functional systems, which can enrich it with the secondary systemogenesis. And natural selection can for these immediate early genes. It is specific for learning. It is rather universal. It involves long-term changes of neuronal properties and required for consolidation of memory. So we decided that we found a marker which can be used for the wide-scale mapping of different functional systems at the time when they're acquired in the developing or the adult brain anew. We also did different comparative studies, and this I include for our colleagues from the clinic to see how this expression of immediate early genes develops in the brain, not in the adult brain, but prenatally or early postnatally. Here is the induction of some pictures on the induction of two genes, C4 and ZIF268 or EGR1 in the immature brain, in the neonatal brain. You can see the areas where either C4 or ZIF are induced at the P0 at the time of birth on the first day, at P1, P2. These are further sections, and how the learning and new experience can give the additional expression patterns which are specific for this. We found, если я уберу вот эту we also found that the expression occurs in postmitotic neurons. So this is colocalization of expression in of C4 and ZIF268 in NON neurons. There is much more expression in the adult brain. Here is the olfactory comparable to the early neonatal brain. But in neonatal brain, some of the immediate early genes are also expressed in neuroblasts. So this is just a brief description of a large study which was carried by in the lab by Anna Ivanov, who is here, Olga Efimova, and Irina Zarayskaya, as well as some further experiments which are described. But what we came to, in order to see the maps, what we first do in the beginning of 90s, we did the digital atlases of the brain and of the rat mouse brain, and started doing serial sections with in situ hybridization and fitting the serial sections in 3D into the brain. But that is a very laborious task, especially taking radioactivity exposure with the film, etc., etc. So in 2003, I started a project which I call the transparent brain, with the idea that we, by the way, passed through several other stages. We did block face imaging, for example. We did alignment of the stain section to the block face, etc., doing video from the cryostat, but this was still very laborious. So I decided that with the development of transgenic technologies, we can take the markers for expression of these genes, combining the promoter of immediate early gene with the fluorescent gene for the fluorescent protein. The challenge was, if we have these mice as a marker mice for these studies, how can we again efficiently map the whole brain patterns of activity at the cellular level? These are just some pictures of Anna Tiunova will tell more. We did a lot of experiments also on chicks, and particularly with imprinting, and this is the 3D reconstruction of C4 expression in the chick brain after filial imprinting. And these are some. Okay, 3D reconstructions. But with the transparent brain, we started developing three types of technologies. First, before we had transgenic animals, we started developing the whole mount staining of large brain samples, or the whole brain with the antibodies, with the freezing and thawing. Alexander Lazutkin, who is not here, did a lot of efforts into this. It is several weeks of the procedure, but you can finally stain the whole adult mouse brain, for example, with the penetration of antibodies. The second was how to clear it, how to make it transparent. And here you can see one of our first brains in 2007, cleared. It is an adult mouse brain lying on the sheet of paper where it is written the transparent brain, and this is before clearing and this is after clearing. And the third challenge was how to image this. And that was the question that even after clearing the brain, if you image it, you will have the compressed image of all the planes. So you need to return to 3D. And we started developing a technique which came from the very old ideas at the beginning of the 20th century, but using laser microscopy, light sheet microscopy. So here are some examples of different clearing solutions which we use. These are different fixations. This is the adult mouse brain. So here you can see, for example, it is nearly completely about 95% optically clear. Then we used the expertise of our physicists to develop the laser sources and microscopy with a very thin, knife thin, 6 microns beam of laser, which can excite fluorescence only at a particular plane. And we developed the light sheet microscope. Here is the one which we used. And this is just an example of the imaging of mouse embryo using this microscope, where you can, with a different density, for example, clear the surface and look under. This is autofluorescence at this in this case. So what was done is that the optically clear sample is placed in the clearing solution, and under the light sheet illumination, it is moved vertically, and we collect the virtual sections taken with the camera. I used for this the just appeared Olympus macroscope with the excellent objective of having NA 0.5, which allows cellular resolution at the scale of 2-3 centimeters. That was our first image of hippocampus in the mouse brain after fear conditioning. So this is the plasticity induced, and there are vessels filled with erythrocytes, autofluorescence, but the green dots here are cells that express C4 after this short single trial conditioning. We did it also with the whole brain staining. This is a usual mouse, young mouse, by the way, without any transgenic technologies. And you can see that it is possible also to see, again, each green dot here is an individual neuron. It was early experiments in 2008. Later, we developed also Olga Efimova in our lab, the Lumos solution, which is water-based and doesn't suppress the fluorescence. And here are some samples which we obtained with this fluorescent imaging. This is the whole mouse brain with the of transgenic mice with the THY-GFP. So you can see here expression individual neurons, and with the larger portion of the same brain just cut, you can see that it is possible at the scale of the whole brain to obtain cellular resolution and to see even the apical dendrites of pyramidal cells and axons stained in the brain with this resolution. We later used different techniques to combine technologies for inducible genetic modification of the neurons, which we can label during the episode of new functional system modification. Here are some data from these experiments. You can do also double staining with the other cell markers, so you can map what cells in the different brain areas are involved into new functional systems and to what type they belong. It allows also of the genetic modifications, which I will not focus in today. Models, the final part. We used the approach of systemagenesis, which was first actually applied practically by Fox in the Jackson laboratory. He used the theory of systemagenesis to map the developmental stages of development of mice in Jackson laboratory, having different periods. So you can see here his depiction of different functional systems when they mature. Later on, Altman and Sadarham used another set of steps to see the battery of different tests to see the maturation of different functions, again, functional systems. And there are some tests to see the ontogeny of different forms of learning and memory. So combining these approaches, we developed at our department, Irina Zarayskaya, Elena Alexandrova, a battery which consists of this set of different stages of development from postnatal day 1 until weaning, and tests for somatic maturation, sensory motor development, different functional systems, behavioral preferences, and learning. So we can use this battery both longitudinally or cross-section to see at different times. And these are some illustrations of the tests that we developed. Which can be used, for example, to characterize the pathology of development using different transgenic animal models or plasticity, etc. And it is very sensitive to this. Part of this test also include Anna Ivanova did it. The analysis of mother behavior, for example, at the moment of birth and first minutes and hours after birth, to see also, we know that at the moment of birth, there is a burst of immediate early gene expression, very strong expression, which goes down later on, but when the organism meets the new environment with the many functional systems to be involved, there is a huge amount of expression of plasticity genes. Another important, probably for our clinical colleagues, type of work which was done by Irina and Elena Alexandrova and their students, is to see how the interaction of mother and care of the mother influence the subsequent development of progeny, using the native mother or cross-fostering with the other mice from the other strains. Perinatal learning, which we also employed into this test, and again using natural models of the contact of the newborn with the mother. The search for the mother and imitation of it by the pattern, looking at different time zones for development of different forms of behavior. Olfactory learning, which is very important for newborn animals. Maze learning, and studying the expression of immediate intelligence to map these functional systems, like here, for example, with the olfactory learning in neonatal animals. So conclusions. Theoretical conclusions. From all this perspective and studies of many scientists in this school of studies across decades in different species, including humans, the organism can be understood as a combination of various functional systems. Организм — это совокупность различных функциональных систем. Each functional system is a unit of morphophysiological integration of the whole organism, peripheral and central. Therefore, the importance of the central regulation also for maturation at the periphery, важность центральной регуляции функциональных систем для созревания всех вегетативных периферических функций. There is no sharp boundary between development. There is sharp boundary between development and learning if it is taken through the lens of the functional system. Learning starts as soon as a particular functional system is consolidated. And experimentally, learning in the prenatal and early postnatal period is accompanied by induction of immediate early genes in the brain. This leads to formation of long-term memory. So protein synthesis inhibitors, other interferences and antisenses can prevent this, so it is necessary. This can be used as a cellular marker of long-term plastic rearrangements in the immature brain. Long-term plastic rearrangements can be used to identify the age of cells, the degree of their maturity, molecular phenotype, and this can be done and used to further explore the activity-dependent mechanisms of consolidation and modification of functional systems in the development brain at the level of individual cells and their populations. Which is particularly important, что особенно важно для понимания процессов пластичности, вовлекающихся в ситуации, например, недоношенного рождения, когда эти функциональные системы начинают модифицироваться на лету, на ходу, и понимание, каковы их ресурсы, как далеко и как много это можно сделать. Thank you for your attention. We probably have, yes, time for a couple of questions.
Talking about functional compartments, when we look at these developing brain and look at even when the sulcus is not formed, and I'm looking at the boundaries, for example, which will be future frontal or parietal, have you done any experiments of looking at the gene expressions, how it is at the boundaries?
No, it is very interesting. I can tell you from other area of our experience or expertise in the chick brain. I started to look for the expression of C4 at ages, and the expectation was that the expression would be in the particular brain area of the avian brain, which was called intermedial medial hyperstriatum ventrale, which they have identified in 70s by different lesion electrophysiological techniques as critical for imprinting. What actually happened was that if you will take this is hippocampus, this is which is called today is IMM. It was called MHV. What actually happened is that the expression during imprinting, I showed it there, it is clearly seen, covers only the portion of the area, but then it goes across the lamina, which are here, and crosses into other areas. So the functional pattern of activation negates the anatomical boundaries. It covers only portions of the particular areas and continues into the other areas as if there are no anatomical boundaries. I called it at that time dorsoventricular reach because it goes in the medial part of the avian brain with its own neuroanatomy.
Mhm.
So I think it is very interesting moment.
Yeah, I would have thought maybe it's like two region functional compartment where we are seeing the boundary structurally. Even in terms of gene expression, there would be like a gradient near the boundary.
Yeah, exactly. Let me show it. You can see it here. You can see it easily here. This is this portion. You can see it on in situ hybridization here, and on 3D reconstructions, it does not correspond to exact anatomical areas, but within itself, as a population, it is rather solid.
Would they not be like parts of the same system that are in different functional areas? So, like in the cortex, you have many cortical areas, and you have reversals and that sort of thing. What you get in there.
That was my idea. Yes.
Yeah, that's sort of thing. It's interesting. I'm a little surprised how you downplay the date of birth a bit.
Mm-hmm.
You know, I agree that things are across learning and things happen across birth, but birth is going to be such a major event.
It is a major event.
I saw some of the things really correlate with birth.
Absolutely.
And others don't, yeah.
Because it is transition from one environment to another critical environment. So most of the functional systems are, I told you there is a burst of immediate origin expression.
Yeah.
They are consolidated before birth. The same will happen with the preterm children at the moment of birth. But this is a massive amount of functional systems which are concentrated between one environment and another, and they start to function. That's definitely. But if you think of this conceptually, theoretically, this, of course, happens with many functional systems here, but this can happen earlier on or later on with the same regularities of transition of primary systemogenesis, consolidation, and beginning of modification. So if you study it in a principled manner, you will find these blocks.
Is there anything different in the systems that are being developed before birth and consolidated at birth, and those that are being developed after birth and consolidated? Is there something like a difference in the complexity of the behaviors associated with these things that can be explained by the change in the environment?
I guess so. The postnatal systems are more variable at the stage of the contact with the environment because the environment is more variable. So they are ready for a wider amount of what they have to expect comparable to homeostatic systems which are. But even with those systems, Irina and Anya did experiments, for example, with the prenatal olfactory learning. You can condition mice prenatally to particular orders.
It will be interesting to see how these systems are in premature,
Exactly.
how this much is
Exactly. We didn't do this, but
I
it
also
is
can't think of
point
how
of
would you
yes.
do it.
Mhm.
How would one?
Well, I can tell you that when I was a child and was visiting the Institute of Physiology, where many of these studies was done in the middle of 60s.
Yeah.
My very strong impression was of the preterm born guinea pigs that were kept in a kind of a taц, как сказать, in a kind of a bowl in physiological solution.
Okay.
So they were born, and the scientists were studying their physiological. There were a lot of electrodes for recording their functions, breathing, electrocardiogram, neuronal recordings from the brain, and they were kept in the special solution, I think, close to physiological. So that was my very strong impression. You can do this, and this has been done at least from 60s.
Yeah, I guess you could do some deliberate early births
Mhm. Yeah.
in an animal model to see if you can imitate.
I think we have to move on.
Yes.
Because we have our next speaker. Anna Tionova, she is from our institute as well, and she will tell about the similar type of research studying the interaction between development of innate predispositions and learning, but using a different model, the avian model.
Good morning to everybody. It's nice to see you all here after yesterday's excellent talks. And our session today is another kind of research, and my talk will be a continuation of what Professor Konstantin told about boundary between development and learning, but in avian species. I would like to start my talk from the citation of a great ethologist, famous for his studies of sign communications, especially in songbirds. And this citation talks about controversy or interaction between nature and nurture, and how can we decide which of them are we dealing with. And Mahler says that there is no chance that we will have understood this in human beings, but with animals we at least have some chances. And one of most suitable approaches to this problem is studies of filial imprinting in the commercial birds. And I will explain why. Filial imprinting is a very specific process in which a young animal develops a strong bond and learns to recognize the parent and follow it. And this is a very special kind of learning. It consists of several different components. Some of them are innate, like tendency to approach moving objects, and innate predisposition to recognize its biological parents. And there is also a learning component, which can be only acquired with experience. And the term imprinting refers only to this learning component of the preference formation. Filial imprinting is a term introduced early in 20th century by German scientists, and different terms used in German and in English all were used to give an impression of imprinting, stamping in, like we had something mild and flexible, and then we just stamp in some impression, so this is this suggests that imprinting is a instantaneous, irreversible process, quite different from other forms of learning. First, very fast, then irreversible, and also occurs within critical periods, and actually don't have visible reinforcement in this learning. So let's, I'll give a very brief review of these points to show you is it so or not, because since 19th or beginning of the 20th century, the views on the imprinting somehow changed. So, in nature, filial imprinting occurs indeed within strictly bounded, sensitive or critical periods. For domestic chicks, this period is 1 or 2 days after birth. What is special about imprinting? I would say that imprinting, the term imprinting can be referred to different processes, but I will tell about a very narrow sense about filial imprinting in precocious species such as chickens, ducklings, and also moose, horses, and other precocious animals. And why is it so useful to study interaction between nature and nurture? Young chicks, for example, are able to see, to move, to peck from the very first hours after their hatching. They are not actually tabula rasa, how it was thought previously, but we at least can control the individual experience and to deal with the brain, which content we are aware of. And the third thing is that they are not only able to learn, they are very eager to learn, because this is from what their survival depends. So first, critical period. Then it was thought that the function of imprinting is to develop species recognition, which means that after imprinting, the youngs will recognize their biological parents, follow them, and form social preferences and social attachment and binds, which were effective during lifespan. And we thought that imprinting is irreversible, and once formed, it will influence all the life of the animal. So, how it was in nature? Filial imprinting in lab is somewhat different. First, critical period we can change by different manipulation and extend it up to several days. Then, after imprinting, the outcome really will not be a recognition of the species, of the conspecifics. The outcome will be to recognize and perform attachment to specific individual object or animal to which the young animals have been exposed. And then last one, irreversibility. After learning on a particular object, chicks can reverse their preferences if they are underwent to exposure to a new one object. But it's not so simple, and I will tell about it. Critical sensitive period is defined by both endogenous processes and the individual experiences of the animal. For example, external factors are isolation. If the chick is kept in darkness without any contact to objects, the critical period will be extended to several days. Keeping in darkness or diffuse light or anesthesia, which inhibits brain development and also extends the critical period, separate presentation of stimulus of different modalities so that the brain is somehow confused and again extends the sensitive period. And there are also endogenous factors such as hormones level, receptors activity, steroid hormones level, and all these factors influence the critical period. For example, if we take a chick which is 4, 5, or even 6 years old, in nature, days old, in nature, this chick, the critical period will be long ago closed. But if we administer it with thyroid hormones, then the critical periods will be opened again. The chick will be imprinted again. Species recognition. As I said, it was thought that the function of imprinting is to develop species recognition and attachment to biological conspecifics. But actually, we can, in the lab, expose chick to an artificial object. For example, this is famous red box, which was used in many experiments, and the chick will form attachment to it. However, there is some kind of species recognition, which is innate component of the attachment. I will talk about it in more details later, but just to show you these funny pictures when the scientists were trying to define which features of the object are important for naive chicks, which don't have any visual experience before it. So it appears that most of them choose real hen, but not this scrambled hen, real chick instead of scrambled one, and a duck with a head over the duck, which don't have a head. However, if they took a stuffed animal, I'm not sure what is it, well, some predator, then there is no difference whether it has a head or not. So there is some kind of recognition. And this is just to show you the experiments where chicks, again naive chicks, were tested for their preference to biological motions. As you can see, there is no objects, but there is a number of dots on the screen, and one of them represents, can you recognize it? What does it represent? No?
Mother.
The chick recognizes it. It's working hand. And this one is rotating hand. The same set of dots, but not biological motion, but just rotating. And this one is random motion, but something which is walking, but it's not a hand. There was also a walking cat. Yeah. And so they compared the preferences, innate preferences for different patterns of motion. And it occurred that chicks strongly prefer walking hand to the rotating hand, but there is no difference between walking hand and walking something, which is not hand, but is performing some kind of biological motion. And the scrambled hand is preferred to rotating hands, and there is no difference between walking cat and walking hand, because it's a biological object, and it makes biological motion. So this is enough for cheeks to form reference to them without any learning.
Хорошо.
This is a number of different objects to which cheeks have been imprinted in different labs. Cylinders, cubes, something complex, even something very simple, like a Kinder Surprise thing, robot, and also images on the screen. And from all these experiments, one of the results was obtained that naturalistic objects are a lot faster and stronger than artificial ones. Here is a rough scheme of the experiment. The cheek is placed in the running wheel, and it can see a hand or a box during training, and then during test it can see a hand from one side and a box from other side, and the number of rotations of the wheel will show us the preference. And yeah, this is, you can see it, not very good resolution, but you will see for a time how it occurs. First, cheek just looks at the hand, and then it starts to run, and it runs faster and faster, and usually the imprinting training session takes about 40 to 60 minutes. And during this, the cheeks runs several hundreds of meters. And if it were sound here with the video, you would hear that the cheek produces very comfort sounds, because even possibility to see his mother and to do something, to move in her direction is very is some kind of reinforcement to it. So after this session, we take the cheek and place it in the situation of the choice between the object to which it was imprinted and not imprinted. But first, I will show you the result of this training session. Here is 40 minutes of training and number of wheel rotation within each 5 minutes. This line is for box exposed cheeks, and this line is for the hand exposed cheeks. So you can see that activity of the cheeks is higher if they are exposed to a natural object. And if we do more prolonged training, which is 3 sessions, 40 minutes each, then in each of these sessions, the activity will be more and more pronounced if the cheeks are exposed to the fold. So the stimulus is more attractive to them than the box, than the artificial. This is images of radioactive in situ hybridization of cheek brain after this sessions, and you can see even here, without quantitative analysis, that to the box, the brain sections looks more dark than with imprinting to hands. So here is the relative levels of expression of C4 on the cheeks of dark controls, exposed to fold and exposed to box. And we can see some contradiction here, because we saw that the activity to approach the stimulus was higher in the fold exposed cheeks, but the expression of C4 is higher in the box exposed cheeks. And so we suggest that the level of C4 reflects the degree of novelty for cheek. So the hand is somehow more familiar for him, though he or she, where though it doesn't have any experience visual, but its innate expectation is more to the fore than to the box. So the box makes more novelty, so more high level of support in the brain. And about irreversibility. This is design of one of our experiments when the chick was trained within 3 training sessions, either to the box or to the phone, and then was tested 2 times, and then was trained in reverse order. The chicks which were imprinted to hand were imprinted to box and vice versa. And again, given to tests. So in this picture, we can see that the preference in the cheeks imprinted to fold and then re-imprinted to box does not change from the hand to box, while the cheeks which were imprinted to box and then imprinted to hand lost their preference for the box. So the reversibility is as follows. If the cheek was imprinted to a naturalistic object, it is indeed irreversible. But if it was imprinted, exposed to an artificial object, it can be re-imprinted and it will be reversed to the naturalistic object. This is another experiment with rather complicated design. I'll just give some details. The cheek was exposed to one of 2 screens in his home cage, and there was one of 3 stimulus moving. And it appears that if the chip was first imprinted to one object and then to another one, all of them prefer the second one. So if both objects are not biologically adequate, then the imprinting can be reversed. So it was about acquired components of filial attachment. And now I will tell about innate and unconditional aspects of it. As we could see already, there is some innate predisposition to non-naturalistically looking objects. And this predisposition is increasing with age. If we take a chick and imprint it to the box, then 2 hours later it will prefer box. Its preference will be more to box than to hand. But 24 hours later, it lost its preference for the box and indeed acquires some preference for the hand. So the result is 0. And the chicks which were imprinted to hand have preference for it, and 24 hours later, they also have more pronounced preference for the naturalistic object. So there is 2 processes which interact with each other and informing their attachment in the young chicks. So take some water. And one of these processes is developmental. It is development of the disposition to approach stimulus, which is similar to the innate representation of the imprinting of the species-specific object. And another one is learning component. They can contradict to each other, or they can support each other, depending on which stimulus we are using. And it was shown in different experiments that